In today's lecture, we will discuss about difference between the prokaryotes and eukaryotes. As during our previous lecture, which was about the fundamental concepts in genetics, in that we have discussed that there is one important fundamental concept which is about the difference between prokaryotes and eukaryotes. So, before going into detail about the genetics, we will also try to understand about the difference between the prokaryotes and eukaryotes. First, as I have discussed in a previous lecture, that there major difference between the prokaryotes and eukaryotes is the presence of presence of cell membrane. As the in eukaryotes cell membrane is present, while in prokaryotes that cell membrane is basically absent, or membrane bound organelles are absent in prokaryotes, while these are present in eukaryotes. So in this tabular form, we will try to understand the difference between prokaryotes and eukaryotes. First of all, if we talk about the nucleus, then nucleus is absent in prokaryotes, or that is present in eukaryotes. Not actually absent, but that as this is not membrane bound circular form which is basically present in the eukaryotic cell, that's why we say that there is absence of nucleus in prokaryotic cells. If we talk about the cell diameter, that there is a relatively small cell from 1 to 10 micrometer in case of prokaryotic cell, while that cell diameter is relatively large in case of eukaryotic cell, and that diameter could be from 10 to 100 micrometer. If we talk about the genome, then usually one circular DNA molecule is present in prokaryotic cells, while multiple linear DNA molecules are present in eukaryotic cells, and we will discuss in detail in our this lecture later onward. About DNA, not complexed with histone proteins in with histone proteins in eubacteria, while some histones are also present in archaea, while complexed with histone are present in eukaryotic cells. Amount of DNA is also very relatively small in prokaryotic cells, while that amount is relatively large in eukaryotic cells. And as I have discussed that membrane bound organelles are absent, or cytoskeleton is also absent in the prokaryotic cells, while these are present in the eukaryotic cells. Here are the figures of the prokaryotic cells and the eukaryotic cell. This is basically the animal cell, and this is basically the plant cell, which is indicating different types of membrane bound organelles which are present within the plant. Here you can see that these are the membrane bound organelles which are present within the eukaryotes. If we talk about from the perspective of genetics, the major difference between the prokaryotes and eukaryotes is the presence of nuclear envelope. A major difference between prokaryotic and eukaryotic cell is that the eukaryote has nuclear envelope, while in prokaryotic cell the genetic material is in close contact with other components, because there is no nuclear envelope around the nucleus. So there is which surrounds the genetic material to form a nucleus and separates the DNA from the other cellular contents. While in prokaryote, genetic material is in close contact with other components of the cell, which is a property that has important consequences for the way in which genes are controlled. Another difference from the genetic perspective is the difference between the prokaryotic and eukaryotic cells is the packaging of DNA material. In eukaryotes, DNA is closely associated with a special class of proteins which is known as the histone protein. These are the histone proteins which are present in the eukaryotes, and which forms the tightly packed chromosome. This complex of DNA and histone, which is also known as the chromatin, that is present in the eukaryotes, while that type of complex material is absent in prokaryotes. Archaea have some histone proteins that complex the DNA, but the structure of their chromatin is very different from that which is found in the eukaryotes. While true bacteria don't possess histones. So that is another major fundamental difference between the prokaryotes and the eukaryotes. Another difference is the chromosomes, number of chromosomes. Genes of prokaryotic cells are generally on a single cellular molecule of DNA, which is known as the chromosome of prokaryotic cells, while in eukaryotes, genes are located on multiple, usually linear DNA molecules, and these are known as the multiple chromosomes. The copying and reading of DNA are therefore simpler process in bacteria, while that is very complex because of the multiple chromosomes. That is very complex in eukaryotes. Eukaryotic cells therefore require special mechanisms that ensures that each of their chromosome is faithfully transmitted to each new cell. This generalization, a single circular chromosome in prokaryotes, though is not very true. Sometimes they have more than one chromosome, which is also known as the plasmid. But that frequently found on other DNA molecule, which is called as plasmid. But that is also another story. That is also not very complicated as in the eukaryotic cells. So in case of eukaryotic cells, you need a special type of mechanism, so all the important genes are transferred very faithfully to the next generation, to the next cell, and to the next generation, so that the traits or the characteristics are passed faithfully to the next generation. So in future lectures, we will try to understand the process of division, which is meiosis and mitosis, in our future lectures. Thank you.